The experiment is about SDS. So, SDS is stand for sodium dialysis sulfated polycaramide gel electrophoresis. To separate protein based on size, the use of a striking gel and dissolving gel. The principle of SDS Page stands that a charged molecule migrates to the electrode with the opposite sign when placed in a electric field. So, material and why we use the material. We will use SDS polycaramide gel electrophoresis SDS page to separate protein of cellular extracted which contain all protein of a cell. The extracted we are interested in come from two CRISPR-edited macrophage cell liner. We also need to include operated control for this experiment. Control increase the reliability and reproducibility of result. Positive control. A positive control is a sample in an experiment that proceeded a known result that compare with the test sample after the same treatment. It is used to control for unknown variables and confirm that all your reagents work. Negative control. The negative control is the opposite of the positive control. The positive control proceeded a known result, whereas the negative control is designed to proceed not respond after the same treatment. The first step for the experiment is increase the heat block temperature to 95 Celsius. Set a micropipette to 30 UL and attach a tip. And save the volume. When finished the tip, close the box. Draw up the wild-type cell culture. Close the wild-type cell culture. Expand the wild-type cell culture into the wild-type cell tube. Repeat the procedure to transfer 30 UL of the pancreas cells culture. Repeat the procedure to transfer 30 UL of the added 4 cell culture. And repeat the procedure to transfer 30 UL of the added 7 cell culture. The tips can use only one time to transfer cell culture to tube. Number 2. Add sample sulfur. And the procedure is draw up 10 UL of 4 SDS sample buffer. And then close the 4x SDS sample buffer. Dispense the 4x SDS sample buffer into the wild type cell tube. Repeat the procedure to dispense 10 UL of 4x SDS sample buffer into the pancreas cell tube. Repeat the procedure to dispense 10 UL of 4x SDS sample buffer into the edit 4 tube. Repeat the procedure to dispense 10 UL of 4x SDS sample buffer into the edit 7 tube. The protein ladder already contains SDS sample buffer. Number 3 is denature the proteins. Move all of your 4 sample tubes to the heat block. Set the heat block timer to 10 minutes and press start. Remove all of your sample tube from the heat block. Number 4 is centrifuge the sample. Move all of your sample tube to the microcentrifuge. Briefly spin the tube and check the balance. And then remove the tubes. Number 5 is load the gel. Place the polyacrylamide gel in the electrophoresis box. Fill the cassette with 1x running buffer. Remove the comb. Fill the outer gel electrophoresis tank with 1x running buffer. Set a micropipette to 20 UL and attach a tip. Draw up the protein ladder. Move the micropipette to the first lane of the protein gel. Expand the protein ladder into the first well. Dispense 20 UL of the positive control into the second well of the gel. Dispense 20 UL of the negative control into the third well of the gel. Dispense 20 UL of the edit for cells into the fourth well of the gel. Dispense 20 UL of the edit 7 cell into the well of the gel. Number 6. Conduct protein electrophoresis. A. Cover the electrophoresis box and connect the red and black cable to the power supply and the lid of the gel electrophoresis box. Turn on the power supply. Set the voltage to 115 volume and set a timer to 16 minutes. Start the power supply. Wait until the time has elapsed. Turn off the power switch. Unplug the electrodes. And the last procedure is inspect the gel. This is the result of the experiment.